In this week's lab, you're studying the anatomy of the nervous system. Please refer to lab exercise 6 for all items detailed in this video. To begin with, make sure you understand what's composing the central nervous system. This is just the brain and spinal cord, whereas everything else will be comprising what's known as the peripheral nervous system, or the PNS. This will include the 12 pairs of cranial nerves and the 31 pairs of spinal nerves you observed this week. To summarize your activities this week, you're going to be studying the nervous system histology, spinal cord anatomy using textbook images and slides, the spinal nerve plexuses using textbook images, brain anatomy using textbook images and slides, and this will all accompany the sheep brain dissection you'll complete at the end of lab. The very first slide we see is a neuron smear, showing you the basic tissue of the nervous system. The largest cells that you see here are neurons, and they are accompanied by neuroglia, as viewed here as tiny dots in this slide. As you zoom in at 400x magnification, you can see a larger image here of the individual neuron. With the neuron itself, you see that it resembles other cells we've studied in the course of the semester. The cell body is more specifically known as a soma with a neuron. And here you can see the nucleus as a light purple ring with a dark stained nucleolus on the inside. You can also see that axons and dendrites are labeled individually, but you will not have to distinguish between them. You're not able to see here with the particular stain that we have on these slides. In addition to this, you can see a magnified view of the neuroglia. Likewise, you will not be expected to know which type of neuroglia you see on each slide. As we continue to look through material in the lab manual, you run across spinal cord anatomy. Again, you'll be expected to recognize this anatomy using this image from your textbook. As you can see here, the spinal cord is divided into regions that resemble the names of the vertebrae you learned earlier this semester. The first eight spinal nerves that come off of your spinal cord are known as cervical nerves. The next 12 pairs of nerves that come off are known as thoracic nerves, followed by 5 lumbar nerves, 5 sacral nerves, and 1 pair of coccygeal nerves. Each of these nerves is going to be associated with a nerve plexus that we will review in a few minutes. But I also want to make sure we point off that the nerves are actually named for regions of the spinal cords, the cervical, thoracic, lumbar, sacral, and coccygeal regions themselves. The base of the spinal cord actually ends before the lumbar region, and that's known as the medullary cone, also known as the conus medullaris. Distal to this medullary cone, you'd actually see lumbar nerves coming from the cauda equina of the spinal cord itself. This means horse's tail and is characterized by large frayed portions of the spinal cord rather than the thick rope-like structure that you would see superior to all of this. You'll also notice the lumbar enlargement, which you see here at around T10, as well as the cervical enlargement that you see between C4 and C5. These are thicker regions of the spinal cord that are predominant in regions where nerves rising to the upper limbs and the lower limbs will originate. The nerve plexuses themselves are responsible for giving rise to the associated emerging nerves that you see here in this far right column. To understand what the nerve plexuses are doing essentially, when you take a look at the cervical region of the spinal cord, you'll see that the cervical plexus is a web-like netting of spinal nerves C1 to C5. These nerves themselves give off a number of emerging nerves, but the largest one that we highlight in the course is known as the phrenic nerve, which runs to the diaphragm. As you take a look at the brachial plexus, this is comprised of spinal nerves C5 to C8, along with T1, and gives rise to emerging nerves such as the radian, median, and ulnar nerves running into the upper limbs. The lumbar plexus is comprised of associated spinal nerves L1 to L4 and gives rise to the very large femoral emerging nerve, which runs into the leg. The sacral plexus, composed of associated spinal nerves L4 to L5, along with S1 to S4, give rise to the large emerging nerve known as the sciatic nerve. As we continue on, you can see a spinal cord cross-section image associated with your textbook illustrations. We're going to associate this with a cross-section histology image that you'll see in just a few minutes. As we take a look at the cross-section of the spinal cord, you notice first off that the spinal cord, much like the brain, is surrounded by layers of tissue. These layers begin with what is known as pia mater directly on the surface of the spinal cord, and then a net-like arachnoid mater, and then most superficial is the thick layer known as the dura mater. Around the spinal cord, you have what is known as epidural space on the superficial regions surrounding the dura mater, and this is surrounded by connective tissue such as adipose tissue. As we look more closely at the spinal cord itself, we can see that there is a distribution of gray and white matter here, giving this butterfly pattern that you see. As we zoom into that on the slide, you can see these components identified between two different images here. Make sure you're responsible. Make sure that you are able to identify all of those components listed in Part 1 and Part 2 of the lab manual. To begin with here, on the posterior surface of the spinal cord, you see what is known as the posterior median sulcus, mirrored by the anterior median fissure on the ventral side. The very centralmost point is known as the central canal, and this is a hollow space that's filled with cerebrospinal fluid. As you look here on the more peripheral regions of the spinal cord itself, you can begin to see the origins of spinal nerves. Right here you can see what is known as the dorsal or the posterior root, and on the other side you can see the ventral or the anterior root. Those two structures come together to form a spinal nerve, and you can designate dorsal versus ventral regions by looking for this large dorsal root ganglion here. This is a collection of cell bodies of neurons that you're going to be studying, and if you zoom into this, you'll actually be able to see the tiny circular cell bodies within this ganglion. If you look at the innermost regions of the spinal cord at the distribution of gray and white matter, you see first off that all regions of gray matter here in this butterfly-like pattern are known as horns: the dorsal horn, the lateral horn, and the ventral horn of gray matter. As you identify them, make sure that you reference ventral horn, right, lateral horn, or dorsal horn, and include gray matter in the identification. Surrounding this, you have what are known as columns of white matter all the way around. Keeping in mind that this is symmetrical, you would have right and left ventral columns, lateral columns, and dorsal columns of white matter. We will be studying the functions of all of these structures in lecture, and you can preview what we're covering there by reading in the lab manual to see what is found in each of these regions. We move on then to anatomy of the brain. The brain, of course, is going to be seen most macroscopically as two separate hemispheres. Here, this would be the right and the left, and they're divided by what's known as a longitudinal fissure. As you begin to study these, you see that they are also divided into lobes: a frontal lobe, a parietal lobe, an occipital lobe, and here on the side, you can actually see the temporal lobe. All of these lobes here make up what is known as the cerebrum. Just inferior to the cerebrum, you have the cerebellum, which also has two hemispheres, as well as the brainstem, which leads down to the spinal cord. As you begin to move through your images of the brain, you'll see a sagittal section of the cerebrum, spinal cord, brainstem, and cerebellum, as you would see here. This is important because you'll be actually applying these structures from the human brain to the sheep brain, which you'll dissect at the end of lab. To begin with here, you can see what is known as the cerebral cortex, and that is the outermost region of the cerebrum itself. This is an outer layer of gray matter with an inner layer of white matter inside. So it's reverse of what you saw with the spinal cord a few minutes ago. You'll see a large column or grouping of white matter known as the corpus callosum, and that's going to be a connection or a communication site between the right and the left hemispheres of the cerebrum. You'll also see here what is known as the epithalamus, close to the back of the region here underneath this corpus callosum. This is most notably viewed by the presence of what is known as the pineal gland, and this is important for producing the hormone melatonin. You'll also see what is known as the diencephalon running here through the more central portion below the corpus callosum, and this is going to be comprised of the hypothalamus as well as the thalamus above it. All of this, including that epithalamus, make up your diencephalon of the human brain. Just below this, you can actually see the pituitary gland. The pituitary gland is only noticeable in some of the sheep brains that you'll be dissecting today, but in many it has been removed when the sheep brains were excised from the skull. As you take a look here at the posterior region, right back behind the diencephalon, you can see what is known as the corpora quadrigemina. This is going to be 4 lumps on the back of what is known as the midbrain, as you can see here, and they're going to be comprised of the superior colliculi, right and left, and the inferior colliculi, right and left. Below the midbrain, you can see the pons, and below that you can see the medulla oblongata. The medulla marks the end of the brainstem, and then will continue as the spinal cord distal to this region. The last thing you can see as you look at this image are hollow spaces that are found within the brain. These hollow spaces are known as ventricles, and they contain central cerebrospinal fluid, or CSF. You're going to have 2 paired lateral ventricles, and then these both lead into a 3rd ventricle, which then passes CSF down to a 4th ventricle, and finally down into the central canal that we viewed earlier when looking at the cross-section of the spinal cord. You'll be able to find the lateral ventricles, for example, whenever you're observing a sagittal section of your sheep brain. You'll be able to pass through a region here above the thalamus into those spaces themselves. So again, it's important to recognize that those are the only ones that are paired with a right and a left section. The last thing you'll be identifying on your brains are the cranial nerves. Keep in mind that there are 12 pairs of cranial nerves. As you begin to study these, you are responsible for knowing their names as well as the Roman numerals that are associated with them. If you are not familiar with how to utilize Roman numerals, be sure to let your lab instructor or your TAs know so that we can walk you through how to use Roman numerals with these spinal nerves and these cranial nerves. Of course, their names, beginning here with the more frontal regions, are the olfactory nerve, the optic nerve, the oculomotor nerve, trochlear nerve, trigeminal nerve, abducens nerve, facial nerve, vestibulocochlear nerve, glossopharyngeal nerve, vagus nerve, hypoglossal nerve, and the accessory nerve. This week you'll be identifying cranial nerves 1 through 6 on the sheep brain, but you are responsible for knowing all 12 of them as viewed on this image that you see right here. Next week during lab, you'll be identifying the functions and the pathologies associated with each cranial nerve. The last slide that you'll be looking at today is of the cerebral cortex. Again, remember, this is the outermost region of the cerebrum, and it's comprised primarily of gray matter. As you begin to view the gray matter of the cerebral cortex, you'll actually see that the cells here are multipolar with a number of different projections coming off of each one, and that just below this you would see the white matter of the cerebrum. You here can see a close-up view of the cerebral cortex cells within the cerebral cortex, and these are known very specifically as pyramidal cells. So again, these are multipolar with multiple projections coming off of the cell body. And beside them, you can still see glial cells, which again resemble what you saw in the neuron smear slide earlier. Instructions for the sheep brain dissection are very important. Make sure you're reading each step in the lab manual to ensure that you're actually making the correct cuts on the correct structures. There are 3 basic parts to the sheep brain dissection exercise. The first, part A, will be you're looking at a sheep brain with thick dura mater around the outside. It's important that you know you should not make any cuts on this brain. This is for observation only. You will identify all bolded terms associated with part A, and then you will return the brains to the designated containers following your observation. After you've done that, you'll then obtain a second brain, and this is going to be a whole sheep brain without dura. Make sure that you first identify all bolded terms in the lab manual before making any cuts. Then when it's time to make your incisions, be sure to know exactly where those should be made. Make sure that you ask for assistance when trying to identify cranial nerves on the sheep brain, as many of them are obscured and break very easily. During the last part of the dissection, you'll actually be making either a mid-sagittal or a coronal dissection line through the brain. Make sure that you ask your instructor which you are instructed to make. Half of the lab will do a mid-sagittal dissection and half of the lab will do a coronal dissection. You are responsible for identifying brains of both dissections, so make sure that you exchange between your lab groups in the lab. You have these images in your lab manual showing you the inferior side of the whole sheep brain, and then a zoomed in region showing you some of the cranial nerves that are there. Here is a superior view of the whole sheep brain. Here is a coronal section of the sheep brain, and here is a sagittal section of the sheep brain. Make sure that you are completing these dissections with your metal dissection trays and disposable plastic knives. All dissected waste must be disposed of in our lovely gut buckets rather than in trash cans and should not be placed in the sinks. Make sure that all trays and utensils are washed and dried following use, and be sure to observe multiple specimens throughout the dissection in your lab. Again, you are responsible for all bolded terms in the lab manual, so make sure that you ask if you can't identify any of them this week. Good luck!